Homeostasis is a word that you'll see a lot in biology, and it's the need to maintain stable internal environments, to maintain the status quo in your body. It could be temperature, pH, water, what have you. But to do that, all organisms need processes and structures that respond to stimuli in a way that keep conditions in the body conducive for life. And the cell membrane is one of those major structures. The cell membrane regulates the passage of materials into and out of the cell. Materials needed for cellular processes must pass into the cell so they can be utilized. For example, oxygen and glucose are continuously needed for the process of cellular respiration. Waste materials from cellular processes must pass out of cells as they are produced. Carbon dioxide is continually produced in the cell during cellular respiration as a result of the intake of carbohydrates and oxygen. Each individual cell exists in a fluid environment, and the cytoplasm within the cell also has a fluid environment. So everything here is aqueous. So the presence of a liquid makes it possible for molecules to move into and out of the cell. That membrane is semi-permeable. Only some things can get through and other things cannot. And we'll look at what that difference is over the next couple of talks. When you actually want to build that membrane, you're going to start with a lipid. Lipids have hydrophobic natures, so they're going to be able to self-assemble into useful structures. In the last presentation, we saw protobionts, which are the basic beginnings of cells, and they're really just a mixture of proteins and lipids that self-assemble into bubbles. Myceliae and liposomes are similar, but more basic in structure. The phospholipid is what the modern cell uses because it is amphipathic, with the polar head and the nonpolar tail. So what that phospholipid will do is it will assemble into a lipid bilayer with the hydrophilic heads outside and the hydrophobic tails inside. And that is going to provide a separation between inside and outside, separating the aqueous environments. So it's more than just lipids. It's more than just a bilayer. The membrane is a fluid mosaic. So lots of different things suspended in this liquid environment. But because of the structure, large and charged molecules need help to get through. Lipids and other nonpolars can get through just fine because a lipid getting through a lipid is simple. But if you have a large molecule or something with a charge, lipids won't let that happen because lipids are nonpolar. To do that, you need to use proteins. Now, those proteins could be transmembrane proteins going all the way through, like an integral protein, or a peripheral protein just on the surfaces. But the proteins are mixed and their functions vary because of that. The hydrophobic parts allow it to attach to the membranes. The hydrophilic parts allow it to act as receptors. So these proteins could be protein channels, protein carriers, receptors. They could function in cell-to-cell recognition, cell communication, cell attachment, what have you. The proteins, if it turns out, determine most of a membrane's function. One of the other structures besides lipid proteins are oligosaccharides. These are sugars that bind to either proteins or lipids, making them glycolipids and glycoproteins that allow for communication if they're attached to a lipid or cell recognition if they're attached to a glycoprotein. A question that I get quite often is, well, what determines your blood type? And it's these oligosaccharides. It's more specifically, they're like glycolipids that are on the oligosaccharides. Now, these will connect into the extracellular matrix where you will see them as proteoglycans because they're more protein in that regard, but they're still structurally similar. Cholesterol is another important component of animal cell membranes. There's a lot more cholesterol in animal cell membranes than in plant cell membranes, but there is some in plant cell membranes. Don't get that confused. But cholesterol acts as a temperature buffer, allowing cells to maintain their integrity at both high and low temperatures. So it's going to keep your cell stable in a wider range of temperatures than would normally be possible. But it's also going to make that membrane less fluid. So these areas called lipid rafts are going to have a whole lot of cholesterol. And so these are going to be very tough sections. Why is that important? Well, it focuses bioactivity. If you have a region that's really tough, it's going to be very solid. That means that the parts that are in it aren't going to flow around as much as they would in the regular fluid mosaic. So they're going to focus bioactivity, allowing a cell to have regions that keep doing their job no matter where they move. There are really two matrices, the extracellular matrix, which we've discussed before, and the intracellular matrix, which is composed of cytoskeleton components. So the cytoskeleton is going to provide structure and support, and it's going to be mainly microfilaments here. But the ECM is going to be made of proteins and sugars that provide biochemical support, movement, regulation, adhesion, communication, a wide variety of functions like that. Looking overall at the actual structure of the cell membrane, you can see that it's composed of a lot of different parts, and each part has its own role to play. It's going to separate the cytoplasm from the extracellular fluid, the inside from the outside. And only by having all of these parts is it able to play its role in actually regulating your body's chemistry, to keep your cell balanced, and to maintain the homeostasis that you need so much.